Hi, this is Eva from PhaBuilder. Let me introduce our manufacturing plant for you.
As you can see now, this is our lab-scale equipment. Inside is our R&D lab. In our R&D lab, we are mainly doing molecular level experiments and also research on optimizing our extraction process. This lab-scale equipment then will verify the results developed from the lab. The equipment includes 20, 50, 100 and 200 liters of fermentation equipment. After the verification, they are then further scaled up into the pilot plant on the right-hand side. This is our pilot plant. Our pilot plant covers all kinds of equipment needed, such as fermenters, sugar supply tank, nutrient salt tank, alkaline tank, installation system, oxygenation system, etc. The pilot plant is actually a scaled-down production line. The strains that we get verified will go to this primary seed tank first, and after a few hours, if the cells grow well, they will be transferred into the secondary seed tank through the pipeline and scaled up. Finally, it comes to the 5-ton fermenter, which is the final station of the pilot test. As you can see, on the right side is our 5-ton transparent fermenter. At present, the inside is mainly our culture fluid, and you can see the current situation and the level of current fermentation fluid. The rotating shifts inside are bidirectional to enable fully stirring, so that the oxygen concentration can be increased to 9 times. Our fermentation is open and there is no need to sterilize. For example, we can directly pour an entire bottle of fermentation fluid into our production system. This is not usual in traditional biomanufacturing companies. Our halomonas can exist in low salt and low alkaline solutions, where other bacteria cannot exist, so we won't worry about contamination. Our mass production relies on these 3 big tanks, the 30-ton self-priming fermenter and a pair of 20-ton tanks. The self-priming fermenters use a negative pressure to take the air in, together with a high-speed mixer to break the gas into bubbles with micron diameter, so that the cells can be fully utilized. These are 2 recirculating water tanks, and our water is finally recycled into these tanks after utilization and re-entered into our production system to reuse. After 36 hours of fermentation in the 30-ton fermenter, all the fermentation fluid will be discharged into the enzymatic digestion tank at the end. In the tank, we use enzymes to break the cell walls and PHA lipid will be released. Before that, we have already accumulated 90% of the product in each cell, so the extraction is very convenient. Finally, we centrifuge the fluid for many rounds and finish the purification for the fermentation product, and the products were discharged into the drying column for drying. This is our finished goods warehouse. Outside the window is our drying column. The product can be received directly from the upstream spout of the packaging machine, which comes from the drying column outside the window.
In order to meet the demands of various applications, such as injection molding objects, straws, coated paper, casting film, blown film, fiber, 3D printing filament, and many others, PhaBuilder has also developed processes to compound PHA powder into modified granules.
After packaging, the product will be sampled and inspected by the quality inspection team. And after being qualified, the label will be printed and allowed to be sold. Qualified products are stored in this finished goods warehouse. Our production capacity at present can reach 900 kilograms per batch for powder.